Hey there! Today we're going to explore the fascinating world of DNA cloning, which is the process of copying specific genes over and over again. Now, think of this process like making copies of your favorite book. Instead of rewriting it by hand, we're going to use a clever biological trick to mass produce it. But in this case, our book is a piece of DNA containing a specific gene that we want to replicate. Let's start with a bit of background. DNA, our genetic instruction manual, is made up of two strands twisted together like a winding staircase. The steps of this staircase are formed by specific pairs of molecules called nucleotides, which we abbreviate as A, adenine, T, thymine, C, cytosine, and G, guanine. Just like puzzle pieces, A always pairs with T, and C always pairs with G. Now, while our DNA is organized into 46 linear chromosomes, bacteria use a simpler circular form of DNA called plasmids. Picture plasmids as tiny rings, like mini donuts, floating around inside a bacterial cell. To begin cloning, we first need to extract the chapter from our DNA book that contains the gene we want to clone. To do this, we use special proteins called restriction enzymes, which act like molecular scissors. Each restriction enzyme recognizes a specific sequence of DNA letters and makes a precise cut. For example, let's say we use an enzyme called EcoRI. This enzyme hunts down the sequence GAATC and slices the DNA right between the G and A. We apply this enzyme to both our DNA and the plasmid. Imagine cutting out a puzzle piece from two separate sheets of paper. Now, we have matching sticky ends that fit perfectly together. The next step is to combine our cutout gene with the plasmid, forming what we call recombinant DNA. It's as if we're attaching our cutout puzzle piece into a new picture. We add an enzyme called DNA ligase, which acts like glue, sealing our gene into the plasmid's gap. This hybrid DNA molecule now carries both the plasmid's original instructions and our gene of interest. Now that we have our recombinant DNA, it's time to put it to work. We introduce this plasmid into bacterial cells, a process called transformation. It's a bit like giving bacteria a USB drive loaded with new genetic information. To help the plasmid enter the bacteria, we expose the bacteria to a brief heat shock, which makes their cell membranes more permeable, like opening a door for a guest. Not all bacteria will accept the plasmid, but that's okay. We have a way to identify the ones that do. Remember how we mentioned earlier that an antibiotic resistance gene was added to our plasmid? This is where it comes in handy. We place our bacteria on a special, nutrient-rich agar plate containing antibiotics. The bacteria that didn't take up the plasmid can't handle the antibiotics and won't survive. But the clever ones that accepted our plasmid will thrive, multiply, and form colonies. As these bacteria multiply, they replicate their DNA, including our recombinant plasmid. It's like a photocopier that's set on print 1,000 copies mode. Each bacterial cell becomes a factory that can produce our target protein based on the cloned gene's instructions. For example, if our target gene codes for insulin, a hormone that helps regulate blood sugar levels, then our bacteria will start churning out insulin proteins. This is precisely how we can produce large quantities of human insulin for people with diabetes. If you found this topic intriguing and want to dive deeper into the world of genetics, check out King of the Curve, where we make complex science topics easy and fun to understand. Don't forget to hit that subscribe button and ring the notification bell so you never miss out on more exciting content. Thanks for tuning in.